Welcome back, folks. This is Brian Booth. Still beautiful here in Silver Spring. Thank you for sticking with us this afternoon. We hope this session's been really helpful to you. We've got a lot of great questions, so I'm guessing it's been helpful to some of you. And we're going to take a little time and sort of do a round robin to try and answer some of these for you. So I'm going to lead off with some questions that we've got about ISR. So some folks have asked whether ISR is needed for pilot studies. And if you do them for one pilot study, do you need to do them for other pilot studies that might be done? So I would say keep in mind, we specify in the guidance that we're looking for ISR in BE studies and ANDAs. We're looking for them in non-clinical studies, once per species per method. And we're looking in NDAs and BLAs for important studies. Okay? So generally, I would guess that pilot studies wouldn't fulfill these criteria. Maybe it does. You're going to have to make a decision about that. But usually with a pilot study, you're trying to figure out what you're going to do in a more definitive study that comes later on. So probably most of the time, you wouldn't really need to worry about ISR and these kinds of things. And that also applies to, like, if you did it once, would you need to do it for other pilot studies? Again, you'll have to make your own decision about that eventually. All right. Here's another question that we got. So our method was validated several years ago in compliance with the 2001 FDA guidance. We're planning to do GLP TK studies soon. So should the method be updated and be in full compliance with the FDA's 2018 guidance? And the answer to that is yes, most definitely. So that takes care of those ones. What else have we got over here? So we had a couple of questions about what are acceptable methodologies for comparing new technology versus conventional technology. So really a question around cross-validation. So earlier today I gave you two examples of how folks do that. And that's about as far as we get in the guidance. We don't specify any particular method. One thing to consider though is most people are doing the case where they're running the samples on both methods and regressing the answers against each other. So you get a linear regression. We have seen cases where you can't always eyeball that. Sometimes there's some bias on the lower concentrations that doesn't show up in that representation. And that can be a little problematic. We've had cases where that has given us some trouble in interpreting clinical studies later in time. So how do you look at that? Well, some of the things that have been suggested is that you do a bland Altman. So if you do that kind of analysis, it really gives you a good visualization of whether the bias is equally distributed across all your data. Some other people have suggested doing Deming regressions, which is also another means to see whether the bias is equally distributed. So those are some of the things that you can look at. What else have we got? So here's another question. It says, parallelism is to be done for endogenous compounds only. This discussion is not covered in the LBA. Well, not entirely true. So we've made a point of talking about, in very general terms, for endogenous compounds. But for the LBAs, if you go look in the table under the specificity and selectivity section, you'll see we refer to checking on the parallelism. And so essentially you should do that either in validation, but more likely when you have some study samples, if you seem to have a problem. Now, what else have we got? What is the agency's thinking on the application of this document to the development of cell and gene therapy products? Well, that's a good question. So the guidance delves into a lot of the details around mass spec and ligand binding assays, a lot of details about that. They may not be applicable to some of these other technologies. So remember I mentioned to you that there are certain questions that we're trying to address. Okay, so you can use this document as a sort of a philosophical approach to try and answer those kinds of questions. But the specifics might be quite different. What else have we got? So next question. Does this guidance apply to things that are being submitted to CDER? Okay. Well, CBER was not a part of this guidance, and so they might have quite different expectations. I know I've heard from some of my friends over there that sometimes they use the guidance for some of the things they do, but it's not a sure bet. What you really ought to do is, if you're going to develop something over there, is to ping those folks and get feedback about what their expectations are for method validation. And I'm going to take one last question, and then I'm going to hand it off. That question is, what group of the FDA reviews and comments on bioanalytical validation for non-clinical GLP tox studies? Okay, so that's part one of the question. So in CDER, in the new drug section for NDAs and BLAs, every therapeutic area has a multidisciplinary team. And the pharm tox folks are a part of every one of those teams. They're the people who are responsible for evaluating non-clinical toxicology studies, including the bioanalytical work. The second part of that question is, what cross-validation is required for different species where the full validation is done in the initial species? And my answer is, I don't know. I don't really deal with that too much, so I can't really give you a good answer on that one. And I think, at this point, I'm going to hand this off to Leah, who's got a few questions for you.
So I'll start with the first question we received. What is the range of the HQC? For example, is there a certain percentage of the ULQ? So to answer that, there is no percentage range of the ULQ for the HQC, as long as the HQC is in the upper portion of the calibration curve. So anywhere between your mid-QC, and your upper limit of quantitation is acceptable. Next question. For the LLOQ QCs, should these be prepared 10% higher than the LLOQ so that all results are at or above the LLOQ, or should the LLOQ QC be prepared exactly at the LLOQ, and we accept results both above and below the validated LLOQ? So I can see the reason for this question. Because the LLOQ is actually a calibrator, how can it also be a QC? It should still be measured at the LLOQ and not at a 10% higher amount than the LLOQ. So the answer to that is the LLOQ should be prepared exactly at the LLOQ. And the next question. For method transfers, you describe a partial validation. What method attributes should be validated and what should not be validated? Well, as you can see in the actual BMV guidance, the partial method validation can range from as little as one intra-assay accuracy and precision run to nearly a full validation. And also, as I mentioned in my talk this morning, if you're extending or truncating your calibration curve, you're also going to have different QCs. So leaving it up to you, as little as one intra-assay A&P run and to nearly a full validation run. You'll have to make your own judgment on that. And the next question I have, in addition to the validation of dilution integrity, should diluted QCs also be included in each analytical run where diluted samples are analyzed? In the BMV guidance, it also says it is recommended if not validated pre-study. So we're recommending that you validate them pre-study, but if you do not, then you will run the dilution QCs in each analytical run where you're reanalyzing diluted samples. And the last question I'll take, what is the opinion for recoveries greater than 100%? We don't really have an opinion, but I think you're asking, will we accept recoveries above 100%? And the answer to that is yes, we have. As long as they're consistent across the 3 QC levels that you're measuring recovery, that is acceptable. And I will now hand off the question and answer session to Suma. Thank you, Leah. So, first question is, what is the purpose of ISR? I think everybody spoke about today the purpose of ISR, but I want to reiterate, it is a means of assessing the reproducibility of the analytical method. Sometimes it can also provide assessment of the stability of the analyte, that is, in case if there is interconversion happening between the analyte and metabolite ex vivo, that you can see from the ISR concentration values. There seems to be a confusion in one of the points I stated that if ISR fail, it should not be the reason for the rejection of the run or the reanalysis of the samples. What do meant is, if ever there are large number of samples that have higher difference in the difference between the original and the ISR concentration values, that is much greater than 20%, either 40 or 50, investigation need to be performed even though ISR fail. However, if needed, you can change the method or sample processing and reconduct the analysis of all the entire study samples using the newly validated method. However, what I meant is, if ever you see any difference in the ISR concentration value higher than 30 or 40 or anything higher than 20%, do not go back and reanalyze the original sample, or do not go back and just reject that particular run or particular individual sample. So moving on to next question. Is a sample dilution should be performed with a biological matrix or assay buffer? For both kind of assays, LBAs and LC-MS/MS analysis, always the sample dilution should be with the biological matrix. Next question. What is the FDA's perspective to reject the run while either LLOQ or ULOQ have failed? CROs shall select the next higher calibrator as the new LLOQ or the next lower point if the ULOQ fails. In some cases, the CROs reject the run. It depends upon what's in your SOP. However, it is acceptable to reanalyze those sample concentrations that are below the adjusted LLOQ or higher than the truncated ULOQ in the case that either LLOQ or ULOQ of that particular run fail. So last question I want to answer is, if the PK data is not as expected per trend and sponsor requests reanalysis, is this a good reason for the reanalysis? There are many questions on this. Some of them ask whether, do we accept the confirmatory repeats, or if the concentration values are not expected in the Cmax what you want to get or in the elimination phase. Usually sample reanalysis with a documented analytical reasons are only acceptable. If ever you see any inconsistent results or incongruent results than what you are expecting, then typically it is considered as a pharmacokinetic repeat. If ever you want to reanalyze, they should be supported by the SOP, and it has to be properly documented, and investigation have to be performed, and also you should provide the reason for these repeats. With that, I will hand it over to Julia.
All right, thank you. This is Julia. I'm going to cover a few questions. So the first one, if an inspection occurs during an ongoing study, to what extent does the inspector expect the documentation in the study file to be available in the file at the time of inspection for study reconstruction purposes? Good question. We understand that for an ongoing study, not all documents or records may be QC'd already or compiled in a study file like neatly in organized manner compared to the completed study. The important thing is for the information. So long as you can provide sufficient documentation to allow the study reconstruction that was done up to that point when our inspectors are doing the inspection there, then that information should be sufficient. It's kind of related to that question, but the second question that I'm going to answer is, where should we include the investigation report for failed ISR? Should this be submitted along with the validation report? And I like to say where, in terms of it should be included, it's not as important as the fact that it was actually well investigated and documented. We, during the inspection, will be flexible, and when we ask the documentation or the investigation report, we just expect them for the form and the CRO to provide the information. Again, where it is less important than all the evidence that we can verify during our inspection. This will be my last question that I'll be answering. During an inspection of LCMS data, what is the objection to PDF as a raw data and reviewing these PDFs? Again, this is another question that we get asked quite often in the past, not as often these days, but I think we've been asked this question before, so I will try to answer it. FDA inspections rely on source data. We look at the source data to reconstruct the studies. If the PDF files are only version available for inspection, they will just make reconstruction of study a bit challenging because of a few different reasons. First, those PDF is having the dynamic nature, interactive nature of electronic data not usable. If you like to have a confirmation of either reintegration or other factors. But more importantly, if you are talking about chromatogram PDFs in this question, there are so much more data than just a PDF. It's kind of called data behind the data, which we call metadata, that PDF will not show. In theory, if anybody wants to print out all the data that we talked about in a PDF format, in a certified format, it might be possible, but that's just not practical. So just looking at and storing the PDF of chromatogram will not be sufficient for us to be able to verify all the data that we wanted to do. We wanted to verify during inspection. Now I'm going to hand it off to John.
Thank you, Julia. So I'll be answering two questions here. The first one is, are there any criteria to select internal standards when you don't have an exact match? So let me be clear here that no, we don't spell out or provide any specific criteria for internal standard selection. You should determine what is suitable for your method, provided that the selected internal standard serves the purpose. Now, you may use literature or other information from your method development. And the second question is it a must to have a LIMS system in the lab to accept the study? LIMS spelled L-I-M-S, that being a Laboratory Information Management System. LIMS is not required. However, LIMS, we understand, helps track data analysis and data reporting as well as sample tracking. Regardless if you're using a LIMS or something else, you should have a process in place that ensures that you can keep track of your data and provide complete study reconstruction. I'll now hand it over to Dr. Panelists.
Thank you, John. My first question is, can you define fresh? And does this mean it's immediately tested, or could this mean within a week? So let me throw out sort of another question, and that is, does fresh mean that everything is freshly prepared? Or does fresh mean that you could be using a stock solution for a period of time and then freshly making the calibrators and QCs with that? And I think the answer to the question is that it depends on what information you have. Early in method validation and method development, the stability data available are limited. So fresh usually means their stock solutions are fresh and everything is prepared fresh on the day of use. It's not uncommon for stock solutions that you would have stability data for those. So it's potentially you can make a stock solution, know that it's good for a week, use the same stock solution, but you don't have information about analyte stability in the matrix, and so you would be preparing them fresh on the day of use. And then as you move along, you develop more stability data, and then you're definition actually changes a little bit, because you have confidence that the stock solutions are stable, the analyte is stable, potentially for a short period of time. So I think the answer is it depends on what is known at the time in method validation. Another question is how can establish a weighting factor model for regression, and what is the importance of a weighting factor? And so usually, as we recommend, the importance of a weighting factor is to try to get the best fit to the data that you can get. The recommendation is to use the simplest model that best fits the data. So if you can do that with a simple linear regression with a simple weighting factor, that's the recommendation. So we never recommend you use a more complex model than is necessary. Another question is, does FDA only accept analytical data from a GLP lab? And if we are following GMP guidances but not certified, are the data acceptable? Well, just to maybe separate what the meaning of GLP. GLP is like good laboratory practice, but GLP really are regulations that describe the conduct of safety studies, and these are non-clinical safety studies. So when you're talking about an analytical lab that's performing sample evaluations for an in vivo BE or BA study, they do not need to be a GLP lab. And many of the analytical laboratories conduct non-GLP as well as GLP work, and the difference is if they were analyzing samples for a non-clinical TK a safety study and doing the TK portion, it's not unusual that those would be analyzed under GLP. So I think what's very important is you got to consider the type of study that you're conducting and whether the lab needs to be following GLP or not. And then another question is if the LLOQ fails to meet the acceptance criteria, the next high level is accepted as the LLOQ. In this case, it would be the CC2 or the second calibrator. So the accuracy and precision for the new LLOQ shall be 20 or 15%. Well, this is one of those where if you were, even in sample analysis, it's not uncommon sometimes to see the lower limit of quantitation QC fail, and in that case, you base it on the CC2. And even though that technically is the lower limit of quantitation for the run, it's not the lower limit of quantitation for the method. So that would be the 15% in that case. And this is similar to another question that says, what about the loss of both the lower limit of quantitation or I guess it's and the upper limit of quantitation standards? Can you adjust the range for that single batch, or does the batch fail outright? But part of it is you got to remember there are expectations in the guidance about the minimum number of calibrators. And I would also say you need to follow whatever SOP you have in your laboratory. So essentially what you're going to get if you have enough QCs, or you have a minimum of 6 and 75% of them pass, so making those assumptions, you would just end up with a very narrow range where you wouldn't, your lower limit would be raised and the upper limit would be low. And then my last question is, if all ISR of one subject failed and investigation showed no reason to reject the original run or the incurred run, and incurred were repeated as part of an investigation and confirm the incurred, can we repeat the whole subject and use the data in the PK in the PK assessment? And I think essentially what this is saying is, if everything is meeting the criteria, so you have at least 67% within 20%, but one subject, and I don't know if it's all 4 ISR samples didn't meet the 20% or not, does that mean you can repeat the whole subject? And the answer is no, but I think it would be concerning, and you might want to investigate why the entire run is meeting the acceptance criteria, but you have one subject in that that isn't. And I guess another question is, what else is happening with the other subjects as well? With some of them failing to meet, 2 out of 4 for those subjects failing to meet the acceptance criteria or not. And with that, that was my last question. I'm out of time, and I'm going to turn it over to Arindam. Thank you.
Thanks, Chuck. So beginning, I will take three very related questions. The first question is, can we exclude single point outliers and present data with and without outliers excluded? When you say single point outliers, if they are due to assignable causes, you can exclude it, but the good idea is to present data with and without outliers. And if there are outliers which you think are outliers, mark it up, make a comment about it. Second question is, do you consider using more than 5 replicates as more reliable results, like I have talked and Chuck has talked during his talk? If you have more than 5, you include all of them in your analysis. Another question is, for A&P validation, all 6 runs have to pass accuracy, or the average of 6 runs can be used to pass accuracy if CV passes. As I stated during my portion of the talk, there are no acceptance criteria for A&P runs. You report everything and include everything in your analysis. Another question I had was, are generic drugs and new drugs evaluated the same way, or are there differences in how these two types of drugs are evaluated? Essentially they are evaluated the same way if they have an analytical component. Another question is the bracketing of subject samples with QC samples be acceptable as an alternative to interspersing the QCs within the subject samples? It is acceptable. You can use bracketing approach, but just look at the trends, how they are functioning. Another question regarding batch. Batch concept applicable only for 96 well plate, not for LLE methods using centrifuges. The example I've shown is for demonstration purposes only, and you can use the same concept for any processing which involves batches. And let's see if I have any more questions. I think this was the last question for me.
Thank you to all of our speakers for your very informative and timely talk, and also for responding to the questions that came in. A few closing reminders. This activity is eligible for 3.5 units of CME, CNE, and CPE. After the webinar, you'll receive a separate email providing you with instructions and completion codes. This activity is also eligible for continuing education by SOCRA, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/CDER-SBIA for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which will be open for 2 weeks. Let us know your comments and feedback via the survey. This will provide us with ideas for future improvements. For questions that were not answered, please resend to cedersbia@fda.hhs.gov. As a reminder, I want to share again how to claim continuing education for physicians, pharmacists, and nurses. Instructions on how to claim your continuing education will be sent to you via email from cedersbia@fda.hhs.gov. Please note, you have 14 days, which is July the 1st, to claim your CE and attest to your attendance. On behalf of CDER Small Business and Industry Assistance, we hope you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CDER-SBIA webinars. Thank you.